Hi, thanks for this great opportunity to present my current project at ISMB conference. I am Yoonkyu Lee, a graduate student at University of Minnesota Twin Cities. Our group is studying acute myeloid leukemia. Acute myeloid leukemia is an aggressive hematopoietic malignancy with a 2-year survival rate of approximately 30%, and more than 50% of patients undergo relapse even after the initial standard chemotherapies induce complete remission. Leukemia stem cells, a rare subpopulation of AML that can self-renew and maintain long-term disease, contribute to therapy resistance and disease relapse. Because current chemo and targeted therapies do not target LSCs, our overall goal is to identify the molecular mechanism of self-renewal in human LSCs and investigate the biological features of LSCs that can be targeted to prevent AML relapse. In our previous work, we used single-cell RNA sequencing to define leukemia stem cells in the LSC-enriched subpopulation of MLL-AF9 and RAS G12V murine model of AML. And we understood that the mechanism that allowed them to cause relapse in our murine model of AML. We also showed that only a subset of LSC-enriched compartment harbors self-renewal capacity. Also, we found self-renewal and rapid proliferations are mutually exclusive features of different subpopulations within the LSC-enriched compartment. For the last, our previous work defined a functionally validated gene expression profiles of self-renewal at the single-cell level in a murine model of AML. Next, we wanted to determine whether human leukemia stem cells express these gene expression profiles that we defined in our mouse model of AML. However, currently available methods to compare single-cell RNA sequencing data, for example, reference-based query cell identification methods, are limited because they rely on arbitrary correlation coefficient cutoffs for their query cell identification. Second, they do not provide a statistical assessment for the significance of the similarities and identification. Therefore, existing methods are inadequate to confidently answer our research question. In my project, I developed single-cell correlation analysis, a new computational method to determine whether human AML expresses our experimentally validated self-renewal gene expression profiles at the single-cell level. SCA is generalizable and can be used to analyze other single-cell datasets. Defining the gene expression profile of self-renewal in human AML at the single-cell level would allow us to identify the human-specific molecular mechanism of self-renewal that can be targeted to prevent relapse and death from AML. I will first briefly introduce how SCA works. SCA uses False discovery rate to estimate how statistically significant those human AML cells express the murine LSC subgroups gene expression programs. We employ permutation tests to derive empirical p-values for similarity significance level of each human AML cell, and then estimate FDR to correct for multiple hypothesis testing. SCA defines statistically significant query cells as putative reference cell identities based on the significance level measured by SCA. The details of the SCAs are follow. SCA first computes all similarities between murine LSC reference gene expression profiles and each of the human AML cell using Pearson's correlation coefficients. This step generates observed PCC distribution for each dataset. The second step is to generate no similarity distribution between the murine reference LSC gene expression profiles and all the gene label shuffled human AML cells in the dataset. The permutation repeats 1,000 times to generate a single null PCC distribution that maintains the cell population structure of the dataset. The third step of the SCA estimates the FDR of each AML cell's correlation coefficients by calculating the number of expected PCC discovered by chance at each level of significance. To do this, we compare the number of observed PCC calculated from real human AML data that have greater than each cell's correlation coefficients with the corresponding random expectation estimated from the null PCC distribution. Next, I applied SCA to a publicly available human single-cell RNA sequencing dataset of 16 unsorted AML samples and 2 sorted LSC samples generated in our lab. Using this approach, we detected cells that harbor both putative self-renewing and proliferating LSC profiles in most of human AML samples in both public and our lab dataset, each with FDR cutoff with 0.2. We define human AML cells that express murine reference self-renewal LSC gene expression profile as putative LSCs and proliferating LSC gene expression profile as putative proliferating cells. In summary, these findings demonstrated that the transcriptional mechanism of self-renewal that we defined in murine AML are similarly expressed in human AML. Next, we ask whether existing unsupervised learning methods can be used to detect putative human LSCs and putative proliferating cells in order to compare SCA to existing methods. We visualize the data using unsupervised learning algorithms such as PCA and UMAP. While SCA-defined putative human LSCs and proliferating cells are enriched in the distinct subgroups, self-renewing and proliferating, the existing unsupervised methods are insufficiently precise to detect putative LSCs and putative proliferating cells from their background clusters. These UMAP and PCAs are representative of the types of results we observe with other existing methods. Next, gene set enrichment analysis was applied to all the data that were tested for SCA. SCA identified putative self-renewing LSCs, gene signatures enriched in gene sets of human LSC self-renewal gene expression profiles derived from bulk samples. This result suggests SCA identified putative human LSCs cannot be identified using canonical unsupervised learning models alone, and we have defined putative human LSCs at the single-cell level, and those putative LSCs express functionally validated human LSC self-renewal gene signatures. In the future, we aim to validate our murine reference profiles in autonormal and malignant tissues that have single-cell dataset. We will also refine our human LSC gene signatures to the human LSC specific by comparing to the SCA results in other tissue types. Using SCA, we demonstrated human AML samples express the self-renewal gene expression profiles that we defined and functionally validated in our murine model. These gene expression profiles define self-renewal pathways that can be targeted therapeutically.